is all about reproduction in plants. So some of this stuff will be a bit of a review from when you learned about plant kingdom, and some of this is going to be new. So in terms of what you need to know from this lesson, we need to know that flowering, which is like flowers blooming, involves a change in gene expression in the shoot apex. So that's what's going to trigger it, is a change in gene expression. And the switch to flowering is a response to the length of light and the dark periods in many plants. So some plants will only flower when they have a lot of sunlight, and some will flower when there's not a lot of sunlight. Another one is the success in plant reproduction depends on pollination, fertilization, and seed dispersal. And you should know the differences between pollination, fertilization, and seed dispersal, but you don't need to know the details. So a lot of the time, I've seen paper one questions where you just need to know the order of these steps in order for reproduction to happen. Another one is most flowering plants use mutualistic relationships with pollinators in sexual reproduction. Methods to induce short day plants to flower at a season. So we'll talk about what these short day and long day plants are and how we can manipulate kind of how they bloom so that we can get them to bloom maybe when they normally wouldn't. Flowering in so-called short day plants such as chrysanthemums is stimulated by long nights rather than short days. You need to be able to draw internal structures of seeds, drawing a half view of animal pollinated flowers, and design of experiments to test hypotheses about factors affecting germination. Okay, so here's a diagram that you definitely would be able, or you potentially may be able to draw or label on a test. So this is a flowering plant. This is an example of an angiospermophyte. So angiospermophytes do have seeds and they have flowers. The other type of plant that also has seeds is a gymnosperm. These are like your coniferous trees, but the seeds are exposed. So the pine cones are the seeds of the plant, whereas these angiosperms, the seeds develop within the plant. So let's see everything labeled. And now let's add the functions of each thing. So plants actually have both male and female organs. So you'll see these filaments right here. And at the very end of these filaments, we have anthers. Anthers is the area that contains pollen. And pollen is the male gamete of a plant. So both the anther and the filament kind of make up the male reproductive portion, and we call that the stamen. So the anther and the filament is called the stamen. The female reproductive part includes this part right here. So at the very top we have the stigma. This is where pollen would end up landing. And it's a little sticky, so it allows pollen to kind of get caught here. It moves down what we call the style. And then inside we have an ovary, which has ovules, which are essentially eggs. And when the pollen moves down, it will fertilize the eggs and become a seed. This part right here is called the sepals, and it's just a covering to protect the developing flower. And then we have the petals, and the petals are there to attract pollinators. And what's actually cool is that the way we see flowers can be very different compared to pollinators, because some organisms have the ability to see UV light, and there can be patterns on these petals that we just don't see because we don't have the receptors that have the ability to see these levels of light. So they kind of act as runways, and you can thank Magic School Bus for teaching me that. And just going back, this whole part right here is the female part. And this is generally called the pistil. So the female part is called the pistil. Once again, this is a diagram that you could see in paper 1, or it could be a diagram that you would have to label in paper 2. So for these angiosperms to reproduce, you have to have this sequence of events. We need pollination, which is the process of moving pollen from one organism to another. So is it possible to pollinate itself, yes, we call that self-pollination. But it's not ideal to pollinate oneself because that would reduce the genetic diversity. Ideally, we want pollen to pollinate another flower so that the genetics are slightly different and there's more variation. So pollination, this process happens due to pollinators, and then once that pollen is transferred to the stigma of another plant, it goes down the style into the ovary where it will fertilize some eggs, and then it becomes a seed. Now that seed then needs to be dispersed so that it can grow into a new flower. And there's many ways that seeds can become dispersed. Sometimes it's just through wind. So whenever we see dandelions, we see those little white fluffy things. These are fertilized seeds, and the wind will disperse these, and then these will hit the ground and then grow into new dandelions. Sometimes it can be through animal dispersal. So sometimes some seeds actually need to be eaten by some animals because their digestive systems will help break through the seed coat to allow it to grow. And sometimes even just water dispersal can help move some seeds around. So in terms of pollination, I mentioned that pollinators are the things that do pollination. And so examples of pollinators include birds, bats, and insects. And they form a mutualistic relationship with the plants. So the plants depend on them for reproduction, and the pollinators are benefiting because a lot of the time they're drinking nectar and trying to get the sugar that these plants produce so that they can, nourish themselves or bring it back to the hives. But either way, both kind of live symbiotically with one another. All right, this next portion is all about flowering and how do plants know when to bloom and flower. And we're going to talk about different types of plants and what causes them to flower during certain parts of the year. But ultimately, floral initiation is caused by the growth and differentiation of the apical cells. So those apical meristem will eventually differentiate into specialized cells, and the cells in the shoot apex change how they divide and differentiate due to changes in gene expression. So one of the things that's going to impact gene expression is light, or the length of the day, and we call this photoperiod. Okay, so the photoperiod, or the length of the day, will change gene expression. So we're going to look at two types of plants. We have long day plants and short day plants. Now there are some neutral plants that aren't really affected by the length of the day, but obviously long day plants are going to be plants that bloom more so in the summertime when the days are longer, whereas short day plants are going to be plants that will bloom more in autumn and winter when the days are shorter. And this whole control of flowering is due to a process called photoperiodism. And its critical factor is not actually the day length, it's actually the night length. So how long is the night is actually what is going to contribute. So before we get into it, we have to understand these things called phytochromes. So phytochromes are leaf pigments which can be used to measure the length of nighttime. So we have two different versions. So we have the phytochrome red, which is considered an inactive form. And when red light hits these pigments, it converts it to this other more active form called phytochrome far red. So during the daytime, you're going to have more light, which means you're going to see more red light during the daytime. So it's in the daytime that we tend to see the phytochrome red get converted to far red. And when it gets darker, there's less red light, and then that's going to convert the far red back into the inactive form of phytochrome red. Okay, so I'll repeat that one more time. So phytochrome red is an inactive form of this pigment. During the daytime, when there's more red light, it will absorb that red light and be converted into an active form called phytochrome far red. So this is going to accumulate throughout the day, and you can imagine that the longer the day, the more sunlight there is, the more of this phytochrome far red we're going to see. At nighttime, without this red light, the far red will get converted back to phytochrome red. Now, I said that the phytochrome far red is the active form, and that active form will either stimulate blooming or flowering, or it can inhibit it. It depends on the type of plant, whether we're dealing with a long day plant or a short day plant. So as I mentioned, day neutral plants are not impacted by the length of day, but short day and long day will. So let's look at these individually. So short day plants are flowers that will bloom when the days are shorter. So when the days are shorter, this means that the nights are longer. So they require the night period to exceed what we call this critical length. So there's going to be a certain time period, and as long as the night time is longer than this particular period, then these plants will bloom. So once again, these short day plants will flower when the days are shorter. So they require longer uninterrupted nighttime. So you can imagine that if the days are shorter, that means you're not going to have as much far red, because with shorter days, there's not as much red light to convert the phytochrome red into far red. And in short day plants, far red actually inhibits flowering. So they don't want a lot of this far red because it will inhibit it. So when days are long, the short day plants will not flower because during long days, you're going to create more far red phytochrome, and this will inhibit. So for these plants to flower, they actually need long nights, because we know in the nighttime, the far red will get converted back into red, and it's the phytochrome red that will actually stimulate blooming. Okay, so I will say this one more time. Short day plants will bloom when the days are shorter and the nights are longer. So short day plants require that the nighttime exceed this critical length. In short day plants, phytochrome far red will inhibit flowering. And remember that normally in the daytime, phytochrome red gets converted into far red. So with shorter days, we're not going to be producing a lot of far red. And with longer nights, we're converting it back into phytochrome red. So the more phytochrome red there is, the more flowering there will be. So long day plants are the opposite. So long day plants require shorter lengths of night and longer days. So in long day plants, phytochrome far red will activate flowering. So once again, you can imagine that when the days are longer, there will be more phytochrome far red, and therefore there will be more flowering. So their night period needs to be less than the critical length, because when we have a short night, that means we're going to have only a little bit converted back into the phytochrome red version. Because in the end, these plants want, in order to flower, they want high amounts of far red, which means they want longer days to convert it into that form and shorter days so that not a lot of it gets converted back. So this is kind of a stretch, this SID and LAD, but you can see short day plants, PFR inhibits flowering. So it says the far red levels must be low to flower, and the night must exceed the critical length, whereas long day plants, the far red levels must be high to flower, so night must be less than critical length. An example of day neutral plants are things like roses, dandelions, potatoes. day length does not impact the flowering. So carnations are an example of long day plants. So now that we know kind of what they need to flower, we can kind of manipulate this. So maybe you want these long day plants to bloom in the winter time. You still want them to bloom so that you can sell them. So normally you can see that in order for these to bloom, we need the nighttime to be less than the critical length. We need that long day. Whereas you see when our nighttime is long and exceeds that critical length, you will not see blooming happening. But if you can interrupt this long period of darkness and give it a burst of light, then it can potentially grow. Because as long as if the night is uninterrupted and exceeds this critical length, it will not bloom. But if you can interrupt it with light, then it's potentially they will bloom. So horticulturists can trigger flowering in these plants by exposing the plant to a light source during the night. Chrysanthemums are short day plants, so they require the nighttime to be uninterrupted and exceed that critical length in order for them to bloom. You can see when the days are longer and the night times are shorter, it will not bloom because the nighttime needs to be uninterrupted and past this critical length. Now, if you were to burst light during, like if the nighttime was actually the appropriate length, but you were to give it a burst of light and interrupt that darkness, then it will not bloom. So during the summer months, April to September, in temperature countries, what's that? Chrysanthemums will not naturally flower. So those longer days, they naturally will not. This is accomplished by covering the plants with opaque black cloth for 12 to 15 hours. For example, place the cover at 5 p.m. So if we do want them to flower during the day, we have to increase the amount of darkness that they experience. So you would have to put them in an environment where you can shut off the light early so that their night period, it does eventually exceed that critical length. So plants need to be covered daily until flower buds begin to show color. So looking at these graphs, you can see that for long day plants, in order for them to bloom, you need low amount, like time in darkness, and we also want high amount of far red. Whereas for short day, we want a longer time in darkness and a smaller amount of far red phytochrome. This is just showing that if you were to, have a burst of light during the darkness, then short day will not bloom because once again, short day plants require uninterrupted long nights. Whereas if you do this with a long day plant, maybe they will start to bloom out of turn. And here, let's take a look again. So here I see that my days are long and my night is short. The night does not pass this critical length, so therefore I'm going to see a long day plant bloom, whereas a short day plant will not. If that darkness is uninterrupted and exceeds that critical length, I'm going to see flowering in my short day plant, not in my long day plant. Now note that the darkness does exceed the critical length, but if you were to interrupt it with bursting some red light, then the short day plant will not bloom, whereas you can actually get long day plants to bloom. Okay, and I'm not sure what this is all about. We're going to move forward. So here's a general summary once again of how this works. So long day plants bloom when nights are short because there's less phytochrome far red is converted. And so therefore you're going to have high levels of far red. And how does this work? So these phytochrome far red will bind to a protein, which acts as a transcription factor. And just recall that transcription factors are things that just increase gene expression. This will switch on genes for flowering. It's a promoter of flowering. Short day plants bloom when nights are long. More far red is converted back to the red, resulting in low levels of far red. Because remember, far red acts as an inhibitor, so we want these levels to be low to cause blooming. And time to reverse transcription and translation. So in terms of nature or science, traditionally conservation efforts were focused on a particular population of species of concern. The mutualistic relationship between flowering plants and pollinators suggests that we should be protecting the whole ecosystem and its biological processes. The saguaro cactus is a keystone species of the Sonoran Desert. Whenever we see keystone, basically it's a type of plant that we can use kind of as an indicator of the environment. It's something that can be very sensitive to a change in environment, and it gives us a clue when the environment is changing. The flowers of the cactus bloom only one evening each year. It provides nectar for lesser long-nosed bat, which is an endangered species, which in turn spreads the pollen from one cactus to another. The survival of both the bats and the cacti are threatened by loss of habitat. By protecting the roles played by the bat, the cactus, and the seed-dispersing animals, we can ensure the future of the Sonoran Desert ecosystem. All right, the last little bit about this lesson is all about seed germination. So this is a very common IA topic. So you definitely want to be able to describe the process of germination. But first, let's actually take a look at a seed and the different components of a seed. So the testa is the outer seed coat that protects the embryonic plant. Then we have this thing called a micropyle. It's like a little hole, and this is where water is going to enter and kind of initiate the germination response. Then we have the cotyledon, that's on the inside, and the cotyledon contains food, stores for the seed, and forms embryonic leaves. And then we can take a look here. So the plumule will be the embryonic stem, so it will eventually grow to become a stem, and then the radicle will eventually become the embryonic root, so it will eventually grow to become a root. So a lot of the time when it comes to seed germination or looking at the rate of seed germination, we can look at the length of the radicle. The longer that radicle grows in a short period amount of time, the faster the germination process. And this is just once again comparing monocots and dicots. So we looked at the cross-section of the stem and the roots, and we saw that the vascular bundles are arranged differently depending on whether or not it's a monocot or a dicot. So some other things are, in terms of cotyledons, monocots have one cotyledon with their seed, dicots have two cotyledons. Leaf veins, so in monocots, they generally show a parallel venation, whereas dicots tend to be reticulated, which is more like net-like. And when it comes to roots, we know monocots are actually more fibrous roots, whereas dicots tend to have a longer and thicker tap root with some lateral branches. In terms of floral organs, monocots will have flower parts in multiples of 3, so we're just talking about the petals, and dicots tend to have multiples of 4 or 5. When it comes to stem vascularization, this is the patterns that we looked at with the vascular bundles within the stem and the leaves. And lastly, pollen. Monocot pollen has a single pore, whereas dicot will have 3 pores. So there's a good little summary of the differences between a monocot and a dicot. So seed germination is kind of the process by which we see basically the plant grow out of the seed. So here's a little time lapse of germination occurring. idea. But you can definitely see that the roots tends to grow a little bit faster because it makes sense that you want that root to grow so that it can absorb that water for photosynthesis to occur. So all seeds need to take in water in order for this to happen. It's kind of the initiating thing that will allow germination to occur, and the water will enter the seed through the micropyle, which is that little hole at the bottom. It also needs oxygen for respiration so that it can make energy and allow it to grow, and it needs to be at an ideal temperature so that the enzymes can function best, as well as pH. Now, depending on the species, there could be light requirements. Some seeds may not require there to be light, where some might. Some might. um, there are some interesting other conditions that some plants require in order for germination. So, some plants do actually require fire because the fire will help, that intense heat will help just to allow, kind of weakens that seed coat and allows the plant to sprout. So a lot of times we do hear about forest fires, and this is a natural process and necessary for a lot of plant reproduction, but it's just when it's dry and these fires get out of control, that's when it can become dangerous. Some require actually freezing, so the opposite. So a lot of time there are some plants where we tend to plant them in the fall so that they have this period of coldness and they become frozen. Um, and it's that freezing that can actually allow the germination to occur. Then there's digestion. So I mentioned this as well, that some seeds require digestion, so the animal enzymes to help to break down the seed coat before it can sprout. Then there's washing. So sometimes seeds can be covered with chemicals that are inhibitors and prevent germination, and you need to wash these chemicals away in order for germination to occur. And lastly, we have scarification. So these are just causing physical damage to the seed coat to weaken it. And this can be literally like physically, like breaking it, scratching it, and sometimes this can be done by radiation as well. Okay, so this is a process you definitely need to be able to describe, and it's the steps for seed germination. So first thing that's going to happen is water is going to enter the micropyle and go into the seed in a process called imbibition. So what the water will do is it will cause activate and synthesize gibberellins, which is a plant hormone, which has been involved in cell elongation and division. Gibberellins are going to cause a synthesis of amylase. And amylase is an enzyme that breaks down starch into maltose. So we see amylase hydrolysizing the starch that was stored in the seed into maltose. And then that maltose will then be further broken down by maltase into glucose. And then the glucose can be used by the plant for cellular respiration, and it will eventually be able to create energy and ATP for it to grow. So I'll do that one more time. So the first step is water enters the micropyle or goes through into the seed through the micropyle in a process called imbibition. This will stimulate the synthesis of gibberellins. Gibberellins will cause the synthesis of amylase. Amylase will break down starch into maltose. Maltose will then be broken down into glucose by maltase. So these are both what we call hydrolysis reactions because we're breaking bonds. And then once that glucose is produced, it will be used for cellular respiration in order to create ATP for growing tissues. Okay. So lots of things that we can kind of link with other parts, other units that we've covered. Okay, so once the seeds are metabolically activated, they have the ability to make ATP from the glucose, then we see germination proceed. So first thing, the seed coat or the testa ruptures and the embryonic root or the radicle grows into the ground to extract key nutrients and minerals. The cotyledon emerges and produces a growing shoot's leaves first. A monocot will just have one leaf, whereas a dicot will initially have two. And the growing plant can be divided into the embryonic shoot, and the stem and developing roots. So you have to be able to, potentially design an experiment to test how different factors affect germination. So remember, there's all those things that we talked about, like temperature, pH, um, oxygen availability, but also whether or not it's in like the environment is salty, because if you are putting seeds in a salty environment, then this might prevent water from entering the seed, and also it could even cause some water to leak out of the seed. And we know water is necessary for germination to occur. So if it's in a salty environment, then it might have difficulty growing. So your independent variables will be the thing that impacts the rate of germination, and you can measure germination by looking at the length of the radicle that grows. The longer the length, the faster the rate of germination. Or we can look at it in terms of like what percent of plants do we see germinating. So sometimes you can get different conditions. So let's just say I'm changing the salt concentration. I will have different salt concentration. For each one, I might have like 10 seeds. And so for each trial, I will have like 10 seeds. And then after my period, I will look and see how many of my seeds germinated out of 10. And that can be another way that you can measure germination. All right, that is it. That is all for topic 9. I hope you enjoyed this, and I guess time to prepare for your test.